The third step in secondary growth of dicot root is the formation of cork cambium and cork. Okay, we have already discussed the formation of vascular cambium, then the formation of secondary vascular tissues and medullary rays. Now this is the third step, formation of cork cambium and cork. With extensive secondary growth, within root, means the development of secondary vascular tissues, much pressure is exerted on the peripheral region. That is the region of primary phloem, cortex and endodermis. So lot of pressure is exerted on these tissues, that's why they get crushed. Pericycle becomes multi-layered and its outer layer functions as cork cambium. This cuts off cells towards both sides cells on outer side get suberized, forming cork, or the other name is phellum. Cells on inner side remain parenchymatous forming secondary cortex, which is also called phelloderm. The cork is impervious to water and protects root interior from mechanical injury and entry of bacteria. Okay. Now, due to pressure from inside, outer layers of cork may get peeled off occasionally. It is only few layers in thickness. Okay. So basically, when the cork cambium is formed, from where it develops from the region of pericycle. Pericycle becomes multilayered and its outer layer serves as cork cambium. Now the cork cambium has activity towards inner as well as outer side. Towards outer side, it is going to form the suberized cells. They are without intercellular spaces. They form cork or phellem. And towards inner side, the cells are parenchymatous. They have intercellular spaces, and the region is called secondary cortex or phyloderm. The function of cork, it is impervious to water, and its main function is protection of root from many mechanical injury or bacteria. Now, whenever a lot of the secondary vascular tissue is added from root interior, sometimes the pressure is so much that the layers of cork, they may also get peeled off, and it is only few layer thick, so easily the outer layers along with the epidermis and all, they get peeled off. Now, the important term is, what is periderm? So, phellem, phellogen and phelloderm together constitute periderm. Okay. Phello means cork, phellogen means cork cambium, and phelloderm means secondary cortex. So, all these three layers, they constitute periderm. Now, we have additional term in relation to the secondary growth, lenticels. Now, what are lenticels? These are pores on the surface of root having secondary growth for aeration. So, their function is gaseous exchange, aeration. How they develop? At places cork cambium cuts off loosely arranged cells with numerous intercellular spaces in between cork. These small portions are called lenticels. Okay, we will show these in diagram. Okay, the last part is annual rings. Now, if we talk about the secondary growth in shoot, in stem portion, then annual rings are quite apparent. We can differentiate autumn wood from spring wood. But when we talk about roots, Since the climate around the roots means the soil, there we don't see much variation, much effect of the environment or the season. That's why there is no proper differentiation of elements. So, we cannot observe annual rings clearly. Okay? So, the reason is, as the climate of soil does not vary much, annual rings are not very sharp in roots like stems. Okay. So, if somebody asks, do you observe annual rings in roots? No, we do not observe annual ring formation while the secondary growth of root takes place. Why? Because the annual rings, that is the differentiation of the autumn and the spring wood, that is only on the basis if the seasonal variations are around that particular organ, whatever is undergoing the secondary growth. Now, if we talk about stems, in them they do face the seasonal variations, because of which the smaller or larger vascular elements they develop during the autumn and spring season respectively. So, that is why we can see the annual ring. But in case of roots, as they are in contact with soil, and on soil the climatic or the seasonal effect is not that apparent as it is in the external environment. That is why much variation in the size of vascular elements is not seen. So, we do not observe annual rings there. So, that was all about the steps involved in secondary growth. Next, we are going to discuss the final structure of a dicot root with secondary growth in the cellular form. So, in order to show the final structure of a root that has undergone secondary growth, we will draw the diagram now. So, you have to show the area of periderm first, okay, and for that we will mark slightly raised line, and in this area draw ruptured epidermis. You have to show epidermis ruptured at few places. This is because of the pressure due to development of secondary vascular tissues in the center. You can show ruptured epidermis at one or two places because we are just showing a part. Okay, so almost up to this level, draw the cells of epidermis. Now, on the surface of epidermis is present cuticle. Leaving the places of ruptured epidermis, rest all over this outer surface you will draw cuticle. Now, we will show few layers of cork. So, mark the dotted line, the area. We have just few layers of cork, okay, and in this area we will make a lenticel. So, the cork cambium with any color you can show. So, during your examinations you can use colored pencils but not pens. Okay, I am showing here with pen in order to make it more clear, but during your examinations you have to use colored pencils only. Okay, So the area of cork cambium, for differentiation purpose we are showing nuclei and you can show 2 to 3 layers and they should be exactly aligned. Okay, make it a point, cells should not be alternate, exactly one over another. Just above it, in this gap, we are going to show lenticell. The lenticels you will make loosely arranged parenchymatous cells with intercellular spaces. So this is the area of lenticels, and the surrounding cells, they are just like cambium, arranged in vertical lines. You will show few layers, 4 to 5 layers of perfectly aligned cells which constitute cork. Similarly on this side also, I will draw few cells. In paper also, if you don't have time to complete the cellular structure, you can show few cells of that layer, but the labeling part should be complete. Okay. So I am going to complete only a small area. Okay, so this is the region of cork. Now towards inner side, we have a comparatively broader area of the secondary cortex. So leave almost double space for cortex, and inside cortex there will be now phloem. Almost same length area for phloem. Okay, and the region of vascular cambium, then the region of secondary and primary xylem. Okay, we will draw one by one. So in this area, the cells are circular, larger in size and with intercellular spaces. This region constitutes secondary cortex. You can show size variation, you can slightly deform cells. In this way, this complete area should be filled. Okay. Now, since the innermost layer of cortex, that is called endodermis, that got crushed. So now we have directly the region of phloem, and in this phloem area we will mark certain areas that will represent primary phloem. So I am showing here 2 patches of primary phloem. The rest of the area should be filled with secondary phloem. So primary phloem elements, simple polygonal elements, they get crushed small polygonal cells. Okay, and surrounding this we have secondary phloem elements, and these secondary phloem elements you have to draw polygonal cells, and few cells should be characteristically large. They represent sieve plates. So fill this area with phloem elements. They may be phloem parenchyma, companion cells, sieve tubes or fibers. In this way you have to fill this area. Again I am saying few cells should be exceptionally large. Now the large cells, you will fill them with dots. That marks the presence of sieve plates. Okay, so this is secondary phloem. Inner to secondary phloem we have few layers of vascular cambium. Now before making vascular cambium, draw medullary rays. So one you will show from primary phloem, and the broader one will be somewhere in between. That will meet the primary xylem. Okay. So divide that area so that you don't draw any cells in this area. Cells here are elongated. Just show their cross sections at different levels. When we reach this area, the star shaped area, now the cells should be polygonal. And as we move towards the center of this, elements should be larger. Again towards the corner of the star, size smaller. Because the central one, they are metaxylem elements and they are larger in size, whereas those towards the corners of the star, they are protoxylem elements and they are smaller in size. So on. Okay. Similarly, here also show single line medullary rays. You can draw them at various places. Now we will make vascular cambium. Inner to secondary phloem, few layers of vascular cambium. perfectly aligned. Okay. Inner to this vascular cambium, we have complete area of secondary xylem. So, see, the secondary xylem should always be more as compared to secondary phloem. So, in order to make secondary xylem, mark few areas where we can show vessel elements. Their cross sections are wider. Okay. Now start making xylem. Polygonal areas. Xylem elements comprise tracheids, vessels, abundant parenchyma, and very few fibers. This way you have to complete this area. This complete area. Now there are two ways. Either you can darken their boundaries in order to show them thick walled. Okay. And the second method is you make them all double walled. So when you double a cell, like this is a cell, double it in a way so that the second line is just close to the first line. Okay. And we have a wider lumen whenever we are drawing xylem. So for all the elements you have to double them, smaller or larger. So this way you will double all the cells. Okay. So this is a complete structure, and similarly see this is also xylem. So just where the cells became polygonal, they should be double layered. So now the labeling part. This outermost layer is cuticle. Then we have epidermis. Next portion is cork. Cork cambium. Next purple colored part is phyloderm, secondary cortex. Then we have crushed primary phloem. In between blue colored, secondary phloem. Vascular cambium. Xylem vessels. This complete area comprises secondary xylem, and the innermost is primary xylem. The rays connecting are medullary rays. So this is a part of TS of dicot root showing secondary growth. These three layers comprise periderm. This is all the tissue of secondary xylem. So these two comprise secondary vascular tissues, and this area is lenticel. It's E, okay, lenticel. So this is about the structure. The cellular diagram, it should be learned properly. So we have outermost layer of cuticle, then single layered epidermis, which is ruptured at few places, and below them we have abundant aerating pores called lenticels. Then there are present few layers of cork, followed by 2 to 3 layers of cork cambium, then abundant phelloderm or secondary cortex. Towards the inner side we have secondary phloem having the patches of crushed primary phloem, and then there are few layers of vascular cambium, and in the center abundant secondary xylem is present and few traces of the primary xylem, which is almost that becomes star shaped in the center, and it is joined to the outer tissue, to the cortex, via the broad medullary rays, and some fine medullary rays which may be single or two storied, they are also present between these secondary vascular tissues. So that is the structure of dicot root having secondary growth. Thank you.